Hi, I'm Dylan Mandel with Nebraska Extension, and today we're on the move. We're trying to hit 5 sites across the state because soybean disease is spreading in Nebraska. All right, we made it to the first stop, and we'll see. We've got wet beans this morning. Japanese beetle feeding. These are tall and they're kind of lanky and falling over. The first field is really wet, but it's looking good. The beans are getting tall and they're starting to fall over, but we are having some frogeye leaf spot. Now we're on the long walk back to the car. All right, off to Haskell. An hour and 14 minutes to Haskell Ag Lab. We just made it to Haskell Ag Lab. We got some reports that there might be some disease on one side of our field, so we're gonna go check that out. And then we're gonna go through several of our other trials and do ratings. Here, I'll show you guys what I see. Just walking out, our weed control still looks excellent. There's a couple of escapes, but we'll grab those. I'll start walking through trials, but thankfully the dew has dried off in this field. I'm almost dry from the last one. We have what looks like some Cercospora in here. You can see this golden color on these leaves. That's not surprising. We had a lot of that in this trial already. Yeah, oh, look at this mottling on these guys. I don't know what that is. That could be a potential viral symptom. Let me pull some plant and look at that. So when I'm wondering if a plant has a viral symptom, I'm gonna look if it's on the new tissue. That's checkbox 1. Checkbox 2 would be if it's not on old tissue. So if we go down that plant, it's not down here. So now let me pull the plant. Just on new tissue. What is it? It's hard to say. It's not been a ton of diversity, but we've been seeing a lot of one thing out here, and that's frogeye leaf spot. It just liked the wet conditions here that we had this year, and it seems to have really taken off. So let me give you some shots of what that looks like. This is a polycyclic pathogen, so a spore started that spot, and that spot has the fungus on it. It's going to make more spores that are going to jump and spread and start new spots. So you can see those here, but you can also see a bunch of really small light spots. Those are likely other starting spores that are causing disease. This whole plot is just loaded up with them. Every plant in this plot, every plant in this part of the field is just covered in these, and they range in how severe it is. But all of this leaf area is lost to disease, so it's not going to be contributing to yield. Now, to look for white mold, we've also been down in the canopies. So Diksha and Bella are way over there, walking through most of our white mold stuff, which is over there. I'm, of course, checking all of these too. Here's what a lower canopy looks like right now. Soil surface is wet. It's humid. There's no wind at all. It's all blocked out. And there's a lot of mushrooms because of that. This one here is not a white mold mushroom. It just happens to be a white mushroom that's down here in the canopy. So I'm crawling through these plots, seeing if we have any actual white mold on plants. It's a good field. We had really good frogeye leaf spot out there. Hopefully that shows up in our uniform trials. Diksha and Bella are down there doing some final sampling. But then we're gonna hit the road and head to our next site in Blair, Nebraska. We're still driving. It's a long drive between all of these first 3. Over an hour each, maybe hour and a half each. All right, we're pulling up to site 3. So we're at a site in Washington County, and up till this point in the year, this has been the one with the worst gall midge with a little bit of frogeye and some Cercospora. We're seeing some bacterial blight or bacterial pustule today. First of all, do you see these wilted plants down here in the canopy? Those are probably gall midge being in this part of the field. So let's get down there and grab one of those. Here's that bacterial pustule that I was talking about, right on this leaf right here. That's gonna rupture with rain and make some more bacteria to spread around, but it's not gonna really take over in this field. Let me pull up some of these wilted plants and let's see what we got. I'm just gonna grab this plant and lightly lean it. Oh, it just came right out of the ground. So it's got fungus on it. It's rotted quite a bit so far. So being on the edge of the field, it's not surprising to see a gall midge plant. Okay, so out at this farm, we're finding something interesting here, which is there's a lot of gall midge that's problematic, but also along with the gall midge, we're finding a fungus that's growing on those gall midge wounds. We see more and more of this, especially in wetter parts of the state. So I'm here with Bella. She is working on...
So we have some orange fungal structures at the bottom of the soybean crown, if you want to show.
Let's see
maybe.
this one.
This one? See, it's kind of growing at the edge where there's wounds.
And you just did a summer research project trying to figure out what this might be.
Yes.
What do you think it might be?
We think it might be some sort of fusarium species.
So we're looking at trying to dial that in and narrow that down a little bit. But in the meantime, you're collecting some of them.
We've collected them, we have isolated some of them, and we're going to do some inoculation tests in the greenhouse.
Yeah, so the base of that is just loaded up. Okay, so we've got 2 more farms we're gonna go to this afternoon still, so we better hit the road. All right, here's our next site. This one, the beans always look amazing. This is just a great field, so we usually don't have a ton to report. But I'm walking out towards the middle. I'm gonna dig around. They got more rain here last night, so we're back in mud boots. I'm just seeing a little Japanese beetle here and then a little bit of frogeye. But Bella just yelled that there's a bunch of frogeye over there, so we'll go see that. Bella, do you wanna show us that frogeye?
We have some, I'll pick it off.
Oh, look right there.
Oh, there you go. Yeah, that's a good one.
That's an excellent one.
And this one has some leaf damage and frogeye.
That looks like grasshopper damage. The Japanese beetles will leave the veins, but I'm not an entomologist. All right, we got one site left. We've seen a lot of frogeye to this point. This is potentially one of the worst frogeye years we've had in Nebraska. We've just had consistent moisture in August, and that really gets things fired up. We also just saw a toad. I'll share a picture here. And at our last site, the reason we've saved it for last, aside from convenience, is it has the worst frogeye so far. So we'll go see if it's continued to progress. This field also has a history of Cercospora, so we're gonna go and see what that's showing as well. This field's got a couple issues. The first just being it got some herbicide burn. We're a little plot surrounded by a bigger field, so when they come through to do that, this got hit with some dicamba at the end, and it's got those cupped leaves on top. So I don't know, this happened several weeks ago, but the plants had didn't jump out of it. It's just the edge varieties that got the most of it. You can see that edge over there is pretty bad off, but these 2 middle ones, these are 4 different varieties. These 2 middle ones seem to do okay. There's also bad frogeye in here, so I'm going around rating all of those right now. Deeksha and Bella are collecting tissue samples. Okay, that's it. That was 5 field sites today, maybe 300 or so miles, several hours of driving, and we're all, I think, ready to call it a day. So we've got another hour or so back to Lincoln. We're gonna head back and get tooled up to do it again tomorrow. There was some disease out there. Frogeye leaf spot was probably the worst thing going on in the state right now. I'd say we're in one of the worst years that we've had in my time here over the last 5 years. There's bacterial things like bacterial pustule showing up. That's gonna be like dark, small spots on leaves. Bacterial blight is still around. Lots of gall midge. And some gall midge is gonna have fungal growths on the bottom of it too. So we're working on figuring that out and learning more about that situation. We're back in the UNL Plant Pest Diagnostic Clinic today, and we've got 2 cases of potential bacterial pathogens on soybean. On the right, we've got the traditional bacterial pustule that shows up later in the season. And on the left, we didn't know what this was at first, but it seemed like there were bacteria in it as well, potentially bacterial pustule just showing up differently. We're gonna show some bacterial streaming here, hopefully, and then we'll try to identify what this is.
Oops. So the way that we look for streaming is we will just take cut a small portion of the leaf, try to get about half of that lesion, put a cover slip on top, and we'll go ahead and do this other one as well.
So you cut that lesion in half so it's open, and now we're basically testing to see if the bacteria are going to rush out of there into this water. All right, so we've got those 2 samples in there. We're looking at the original or the more common bacterial pustule symptoms, this leaf over here on the right first. So all of this coming out right there, it's just oozing out of that cell. Oh yeah, you can see the flow.
And so this one is streaming weekly, but if we go and look at the atypical one, and look at all of this streaming.
So can we tell exactly what bacteria it is from the streaming?
We cannot. Can get some hints based on what we know about different bacterial groups and how well they stream, but the only way to confirm whether or not this is a Xanthomonas or a Pseudomonas or any of the other bacterial diseases will be to grow it out on a petri dish and then test that pure culture.
You have some bacteria right here as an example. So a fungus would look dry and hairy under a plate. Bacteria under a plate just kind of looks snotty, oozy. So that'd be a difference. The right is a fungus, the left is a bacteria.
Couple of fungus on the right.
Yeah. Okay, so the next step is we'll take some of that bacteria, we'll put it on plates, and then we'll wait a couple days, and we'll see if we can grow that out and identify it.